Good evening, my dear students. Hello. Ajay, can you hear my voice?
Sir, you are audible, sir. You can
Okay, ma'am.
continue,
Thank
sir.
you. Okay, ma'am. Thank you. Good evening, ma'am. Okay. Good evening, my dear students. Today, what we are going to talk about, evolutionary part 3. Already we saw it. What are the theories? Lamarck's Darwinism and also Hugo de various theories. Already we saw it one by one, isn't it? So, for example, inherited of the acquired characters. What is the meaning of inheritance? The genetic informations and characters also transfer from the one generation to another generation, isn't it? That things already we discussed. This was disproved by the name of the scientists already in our the Weissman. Okay, how the experiment are conducted by the scientists already in our book. And then Darwin theory of evolutions. Okay, which book? Natural selections. In the which year? At the same time, the origin of species and also the by means of the natural selection. These are the important things. Okay, already saw it, the mark and so on. So. And what are the principles of Darwinism? Overproductions, limited and space, struggle for existence. It's also further divided to intraspecific competition and interspecific competitions, and environmental conditions and various among the individuals, and also further divided into continuous variation and discontinuous variations. That things also already we discussed, isn't it? And what are the causes of variation? Already we discussed. There are two factors. Is there? One is called as environmental factors. Another one is called as genetic factors. These are the two things already we discussed. And the most important concept of the Spence survival of fittest. It is also very, very important for the evolutionary criteria, isn't it? Okay, that things also already we discussed. And Darwin's also, what is that? Published the book, okay, in the 5th editions. That things also stated the statement. Okay, which plant Hugo de Vries analyzed the mutation theory? Theoria Lamarckiana. Okay, that is also very, very important. Okay, and what are the salient features of the de Vries? That things also we discussed one by one. Okay, new species originated as a result of large discontinuous variations. Same variation may occur in the large number of individuals. Mutations are recurring again and again. It will be continuously going on to the next generation. Wise. And mutation takes place in all directions. It is not a unique. Okay, it may the all direction happens. That things also already we saw it. Next one is very, very important. That is Hardy-Weinberg principles. This is based on their allele frequencies. Suppose each and every DNA molecule also made up of more than thousands and millions of cells. Okay, each and genes. Each and every genes it consists of which one? There is a locus. Is that the locus consists of alleles? The alleles decide their characteristics of the genetic, organisms, isn't it? This is called as what is that? Based on their gene information or genetic pools or gene and their alleles in the populations, particular entire population, it can be represented. That is called as a genetic equilibrium. It may be represented as a traditional equation is called as a p plus q whole squared, that is equal to p squared, and q squared and 2pq that is equal to 1. Okay, suppose the variations, the value is here is a 1. Suppose if it is vary, what happened, due to the mutation, otherwise called as evolutionary aspect. Mutation, how it will happen, do you know very well? Due to the factors. What are the factors it may be? There are 3 factors have been reported by the evolutionary process, otherwise called as mutations. What are they? Physical, chemical, and biological. We will study on the oncology, otherwise called as cell divisions, like that. No, that time we will study these things. And then what are the factors affecting Hardy Weinberg equilibrium? That means we are getting the value 1, isn't it? How the value will be affected? What are the factors involving? We are going to discuss. See, first one is called as gene migrations, otherwise called as migrations. Okay. And second one is called as genetic drift. And third one is called as mutations. And the fourth one is called as genetic recombination. Genetic recombination means, what is the meaning of recombination? It is also mingle with the foreign particles or alien species. Okay, the genes are, what is that, combined together, as a result form is the new one, that is called as a offspring. Okay, and natural selections can lead to, so what are the things? That things we are going to discuss one by one very deeply in this session. Okay, see here, first one is gene migrations, otherwise called as migrations. Okay, when migrations of section of population. Okay, take an examples. Any one of the populations, what happened? They can migrate from the one generation, one place to another places. That is very, very important. 2, another place occurs, gene frequencies changes in the original as well as in the new populations. Suppose take an example, some of the peoples are, we are all are migrate from the Central Asia, isn't it? So we are all are, which one, new population compared to the Central Asia peoples. That is gene migrations. The new genes, all alleles are added to the new populations. The new genes are alleles. That means comparison between the ancestor as well as the which one, migrating peoples or migrating any organism. What happened? The gene sequence as well as the alleles also may be changed due to the environmental factor, food, habitat, whatever it may be. That is given here. The new genes alleles are added to the new populations, and these are lost from the old population. Okay, that means what is the meaning? The previous or ancestor, they do not have the new genes. That is the result. New genes only present the new population only. Okay, so that is called as gene migrations. See here, the gene migration occurs many times, is termed as gene flow. Let's say after the new populations, for example, the ancestor is the, okay, the ancestor produced a new gene or new population or new organism, like that, after the newly added alleles or genes, isn't it? So the offspring is a new one, isn't it, compared to the ancestor? Okay, it continuously going on, that condition is called as gene migrations occurs many times, is termed as a gene flow. Note it down, this is very, very important point. What is by gene flow? Sometimes they may ask the question, genes are transferred from many times and many generations. Okay, that condition is called as gene flow. Okay, this is very, very important. Okay, see, this is the examples of gene flow. See, this is the ancestor. Okay, this is the brown color. What is the pedals? Okay, present here. These are all ancestor. Here, the one, what happened? One population, the particular population migrate from the one place to another places. See here, this is a green color nature. It comes to here. What will happen? The gene migrate to here. What happened? This is called as new species. This one is called as, what is that? What one? Older one, otherwise called as ancestor. After few generations, what will happen there? This character will be transferred from, what is that, among this themselves, among this organism. As a result, the more population, what happen, in which color? Brown in color in this area. See the next slide, you can understand easily. See here, See, these are the red, isn't it? Red, just imagine compared to the next previous slides, the red is which one, ancestor like that. It migrate from here, what happened, a new production will be happen. Otherwise, new population will be happen like that. Imagine. After 10 or 100 generation, what will happen? There is a mixation of two genes, isn't it? The brown color genes also present, or alleles are present in the green color. What is that? Alleles, isn't it? That is very, very important. See here, the transfer of alleles into or out of the population due to the movement of fertile individual or their gametes. Genetic variation is not only take through the, which one, transformations. Transformation, very, very important. The genetic information, take an examples of bacterial conjugation. Which one is considered as a F plus? Which one is considered as a F minus? You know what is my conjugation, my dear students? What is my conjugation? Hello? Conjugation is nothing but one of the reproduction of which one, bacteria, isn't it? When it's come and contact close together, as a result, the formation of a tube-like structure is called as canal. That way, what will happen, the genetic material transfer from the F1 plus F plus to F minus. As a result, what happened, after the transformation of the genes or alleles, genes only, what happened, F minus is also converted into F plus. Again, the cycle will be continued. This is the process. Okay. Likewise, the genes transfer. This is considered as a gene transfer from the one population to another one. As a result, what happened? It will be produced a new one, isn't it? So that is given as an example. For example, movement of pollen by wind or insects, movement of human around the globe. So this is also another thing. Here the animal has given, for example, migrations, gene flow from the one population to another population. Here what happened, that gene sequences also present in here. After a few generations, what happened here? It is also mingled with the second one, isn't it? Likewise, some of the pollen grains also transferred by wind from 100 meters or 1000 meters from here to other country also. Take an example of border of Pakistan or India and China and so on, whatever it may be. This side also take an examples. Okay, they can, what is that? Spread through the wind. Okay, so it is also another type of gene flow. That is very, very important. See, next one is called as genetic drift. These are all very, very important, my dear students. These are all very, very important. Genetic drift, gene flow. What is my gene flow? How it happens? Like that, they may ask the questions. Sometimes they may ask the fact of questions, or sometimes they ask the direct questions. Okay, and assess and reasoning. Like that, they may ask the questions. Genetic drift, the change in genes. Okay, frequency takes place by chance. Suppose when it gets a favorable condition, it can be changed into what is that? Some other forms. As a result, what happened, it is also here. What is that? They may ask the question, what is in by genetic drift? Genetic drift, nothing but the change in the gene frequency takes place by chance only. Sometimes the chance in allelic frequencies is so different than new sample of population that they become a different species. It is also compared to the genetic gene flow. That means ancestor is also having the gene sequences, separate gene sequences. As a result, new one also has a separate gene sequences. Suppose if it is migrated from the one place to another place, what happened? No, the new one is possible. Okay, but that is one of the chance. Okay, change in the gene frequency takes place by chances. See, the original drifted population become a founder species, and the effect is called as the founder effect. You must note it down. Very, very important. Parent is considered as a founder species. Which one is the original drifted population? That is the parental one. It is considered as a founder species because the presence of which one, new gene. from the which one, ancestor, ancestor to offspring. What happened, the allele also change in the offspring. That offspring is called as what is a founder species. Okay, and the effect is called as founder effect because the allele sequence is changed due to chance. Okay, this is very, very important. So many times they ask the questions. The original rifted population became a founder species, and the effect is called as founder effect. Okay, see that? So this is called as what is that? So these are all are the ancestor, founder species. Here, founder effect. See that the newly founded population don't always represent the genetic diversity in their sources. So these are all are considered as a founder effect. This one is considered as a founder. What is that? What is that? Founder species and founder effect here. Okay, so you must note it down. This is also a very, very important things. And next we will pass on to the next one is called as mutation. Will you please tell me what is my mutations? Already given here. The sudden changes takes or occurs in the chromosome level or gene level or allelic level. As a result, what happened? The new genes are produced. Before two gettings, already we studied the same sequences, same concept. Okay, but here, what happened? There, the genetic information transfer from the one gene to another one due to the chance, or it may be transferred to the one population to another population. But here, what happened? A mutation takes place. What are the abnormal mutations? Already studied it. Addition, deletion, substitution, transfer, inverted, like that. Already studied it. Okay. So likewise, the sudden changes also occurs due to the so many factors is there. As a result, what happened? The DNA alleles, nucleotide. Okay. The sequences will be changed. So as a result, the offspring will also a new one compared to the parent. So the genetic mutation is a chance to a gene's DNA sequences to produce something different. That is called as new one. The genetic mutation is a change to the genes of DNA sequences. DNA sequences mean what is my DNA? DNA is nothing but the genetic material. It is also made up of polynucleotide sequence, isn't it? Please tell me, sir, DNA is made up of what are the three components? DNA is constructed by the three components. What are they, will you please? Hello. Hello, my dear students. Can you hear me? Okay. DNA sequences to produce something different, new one. The advantageous mutation lead to the new phenotypes and over few generations result in the speciations. So as a result, after the DNA sequences, what happened, it may be expressed in the phenotypical aspects. Physiological effect also expressed. So the new phenotypes. Okay, see the next one. See, these are the normal DNA nucleotide. Already saw here. It's one of the DNA molecule is there. See that adenine, guanine, and cytosine and thymine. These are the nitrogen bases also present in it. That's why I asked the nucleotide. And also sugar, deoxyribose sugars and phosphate molecule. These three combinations is collectively termed as a which one nucleotide. When it mutated, see here that some variation is here. Instead of this yellow or say blue, some mutated. As a result, what happened? And the new one is also formed. Okay. So double standard helical structure of the DNA is which type of whether positive or negative rotation? Will you please? Ajay. Dhanalakshmi, can you hear my voice?
Yes, sir.
Why you didn't answer me, pa? Guys, the DNA double standard helical structure, who proposed a Watson Crick already you know very about. It is whether positive or negative. Okay. See that here the another example has given the bacterial transformations. Okay, the bacterial transformations. So this is also considered as a one. This is also another one. Otherwise, it's called as F1 is a donor cells and F2 is a recipient cells. Here, what happened? You know, there is a nucleate DNA molecule is also present in. Otherwise, called a genetic material is there. Okay, it transfer from where to where, you know, in one place to one bacteria to another one. Okay, it release the what is the genes. See here, antibiotic resistance, because it is the absence of antibiotic resistance. That's why. So as a result, what happened? The gene is also transferred from the one to F1 to F2. So what happened? The DNA molecules, what is happened? It is also mingled with the recombinant with the another one. As a result, the new species also formed, isn't it? It is also here compared to similar to that of the donor, isn't it? Once again, what happened? It also undergo the further what is the replication process, or so many times or so many generations. What happened? It also enter into the another cycles like at the previous one. Donor cell. See the next one, bacterial transduction. What is my bacterial transduction? It is also done by the bacteriophage. See that bacteriophage is here. There are three stages there. Adhere on the surface, penetrated, and introduce the nucleic acid. The bacterial nucleic acid here, just imagine, it's enter into the DNA molecule and also penetrated or recombinant with the bacterial DNA and produce themselves as a viral along with the viral DNA molecules. After the formation, already in our that it's cycles, lytic cycle, bacteriolytic cycles, isn't it? As a result, head and tail and hooks are formed and nucleic acids also introduced. As a result, what happened? Complete entire structure of the bacteria was produced. After that, cleave the cell wall of the bacteria, it comes out. And also it entered into another cycles. This is also another type of genetic transformations. And the bacterial conjugation, already I told you. Okay. See here, this is called as what is the extra chromosomal body. The extra chromosomal body, it has a resistant genes. That is very, very important. Already told you PP322 is one of the plasmids present in the E. coli bacteria. It has a resistant characteristics. Resistant genes also possess. Okay. Suppose if it is possessed, it is considered F plus. Okay. So this one is called as F minus. After the transformation of the genes from the F plus to this one is called as donor cell. This side fully is donor. This side are recipient cell. So the plasmids received by the F plus to F minus. As a result, what happened? It is also converted into, that means F minus converted into F plus. So here the transformation of genes. These are all of one of the mutations. Okay. See that very, very important things. Okay. And genetic recombinations. What is my genetic recombination? Nothing but the DNA molecules. Suppose if it is broken, okay, by so many nucleases. Will you please tell me, during the genetic recombinations, which area the gene will be broken by the enzymes is called as what? When the fragment happen, you know, that is a molecular scissor, one of the molecular scissors used for the recombination technology. Otherwise called as trans recombination. Oh, yes. What is that? Will you please? What are the enzymes, Ba? Which enzymes are essential for the? It's act as a molecular scissor. Which one act as a molecular scissor? Anybody else? Dhanalakshmi, do you know?
No, sir.
What about others? Not answering. Ajay Saril?
Sir, no, sir.
So what about the DNA polymerase enzymes? DNA restriction endonucleases. What about DNA ligases? Okay, these are all very, very important for the recombination technology, isn't it? DNA polymerase is an enzyme. Where the cut is mad, the one of the polyandrogenic genes. Okay, just leave it. That things is not here. No need to here. Just note it down. Genetic recombination. Genetic recombination is nothing but recombination. Recombination again and again. It will be united with the host DNA as well as the alien species DNA. See here, the union of two genes from different sources. That is very, very important. Recombination, the process by which DNA stand or broken and repaired, producing a new combination of alleles. When it broke by the enzymes, it is act as a molecular scissors. Polymerase 1, polymerase 2, polymerase 3. Restriction endonuclease also present in it. Okay, restriction endonucleases, what will happen? It made a cut inside the nucleotide. Okay, and repair after the introduction of the foreign DNA and attached with the which one host DNA. And finally, what happened? You have to fix the DNA with the help of the enzyme is called as DNA ligases, linking of what is that genes. Repaired, producing a new combination. As a result, you will get the new genes. Occurs nearly all the multicellular organisms and has important implications for many evolutionary process. Okay, the evolutionary process mostly done by the DNA molecules only. And also the mutation also same. When it mutated, that means introduction of the new genes or alien species to the host cell. As a result, what happens? See the process by which a DNA stand or broken with the help of the enzymes. DNA restriction endonucleases. And repaired. What is the meaning of repaired? It is adding of the nucleotide, whether it may be an addition or deletion. Okay. As a result, what happened? You will get the new combination of alleles. Okay. So new combination you will get, it means the phenotype is also what happened. It is a new one, isn't it? See here, during the gametogenesis, variation due to the recombination result in the new phenotype. We are all the new phenotype during gametogenesis, isn't it? Male and female gamete come in contact close together, and male gametes also have a different types of genetic informations, and also female gametes also it has a different type of genetic information. Both are combined and contact close together. That means when it undergo the fertilization process, zygote formation takes place. That means what happened inside the DNA crossing over and all those things happen. So we are all our new one. Okay, that so the phenotype characters also differ. That is, things have been reported here. A new phenotypes during gametogenesis, variation due to the recombination result in the new phenotype. Okay. Then the last one is called as natural selection lead to. So here you must note it down. Two things is very, very important. One is called as character value. Another one is called as, what is that? I will tell you. Okay. Next. Stabilizing in which more individual occur means character values. What is the meaning of in which more individual occur means character value? I will tell you. Give an examples. Directional changes. That is more individuals or occur value other than the which one character values. See here, next one. What is by a character value? The character value in biology, any observable features or traits of an organism, whether acquired or inherited. Okay. In biological, any observable feature of traits of an organism, whether acquired or inherited. In the inheritance takes place with the help of which one DNA molecules only. An acquired character is response to the environment. An inherited character is produced by the genes. Acquired character means, what is the meaning? Already you know about. Take an example of pepper moth. Okay. At the time, what happened? Only they are eating the seed eating or insect eating. That is depends upon the what is that? Seed eating converted into insect eating. That is the character, acquired character. Inherited character, suppose inherited means what is that meaning? The peak changes and so and so. It is depends upon the genes. See here, the character already told you. An acquired character is the response to the environment. An inherited character is produced by the genes transmitted from the parent to offspring. Their expression are often modified by environmental conditions. This is called as what is that? Acquired character. Genes also. That's the result I told you. Seed eater and insect eater. How it will be seed eater converted to insect eater? But it is nothing but the environmental factors, conditions, or adaptation, radiation, isn't it? After that, continuously it happens, common generation and 1,000 or 100 generation it happen, So gene transmitter. That is called as what is that? Inherited character, because it is and the character will be expressed, character will be transferred from the one generation to another generation, inherited. It only takes place in the gene level. It is the environmental level. That is the very, very important things. Okay. See here. One gene may affect many characters. It is also another one. Some of the plant or animals, a particular gene may affect the many characters. One character may be controlled by many genes. This is also very, very important. Just note it down. One gene, one enzyme hypothesis. Who stated the statement? Anybody else? One gene, one enzyme hypothesis. Do you know that one?
No, sir.
Okay. One gene, one enzyme hypothesis was done by one of the scientists called as Piddle and Tatum. By the one of the fungi is called as Neurospora. These are all very, very important for the, which one the genetic engineering. Okay, one gene, one enzyme hypothesis. Once again, repeat for you. One gene, one enzyme hypothesis. That means one gene produces only one type of enzyme only. Okay. Here, what they told, one gene may affect many characters. A particular gene, they may be control or affect what happened? So many characters. Okay. Sometimes what happened? One character may be controlled by many genes. These are the contrasting, antagonistic effect. Okay, opposite. Okay, this is very, very important. See that a character controlled by only a few genes is known as the oligogenic genes. Very, very important. Otherwise called as qualitative characters. Note it down. What is by oligogenic genes? The oligogenic genes is nothing but the characters controlled by only a few genes is known as oligogenic or qualitative characters. See, the oligogenic genes demonstrated discontinued variations in which the triads are discrete example blood types. Okay, otherwise called as multiple alleles. The multiple alleles. Okay, see here how many multiple alleles means nothing but the blood grouping, isn't it? Who discovered blood group? Who discovered blood group? Who discovered blood transfusion? Anybody else? Multiple alleles. What is the meaning of multiple alleles? A particular locus, it contains more than one alleles present in the one single locus is called as multiple alleles. Okay. It is also an example. So what is that? Oligogenic or qualitative character, because it's produced four different types of blood groups or blood types: AB, A, and B, and O. The blood group was first introduced by the name of the scientist. First discovered by the name of the scientist is called as what is that? Blood group. Landsteiner. Okay, the blood transfusion. William Harvey. These are all very, very important when you are going to study the hematology. At that time, you must understand this concept. Just once again repeat for you. One gene may affect many characters. One character may be controlled by many genes. A character controlled by only a few genes is known as an oligogenics, okay, or qualitative characters. Oligogenic characters demonstrated discontinuous variation in which triads are discrete. Example, blood type is either AB or A or B or O. So this is very, very important. So oligogenic genes may be given examples, means they may ask the questions, multiple alleles. Sometimes they may ask the alternative form, qualitative characters. Okay, which one? You must note it down. See that. A character controlled by many gene is called as polygenic. That one is called as oligogenic. A character, simply note it down, a character controlled by many genes. Or quantitative. There, what is that? See, qualitative. Qualitative means what is that? Few genes, quality. Quantitative means what is that? Numbers. Know the difference. Quantity, quality. Okay. Good quality, good quantity. So 1 kilogram is a good quantity. Okay. But 100 gram, but it is better. That is quality. Okay. Such characters demonstrated continuous variation. Example, weight or height, each of which spans of range of values, lifespan. Okay. So polygenic demonstrated weight and height. Okay. Here what happened? A character. That is a weight is one of the character. Height is a character controlled by many genes. This is called as polygenic. Okay. So this is also a important things. See here, there is a one graphical representation has given here. The number of individual variations with the phenotypes, what happened against the trial. Okay. So the plotted against the number of individual with the phenotype and also trials. There is a plot, you will get it like as a bells like structures. Which one is a constant? This is called as phenotypic favored by the natural selections, natural selected components. How it will be? It may be expressed 3 ways. See the first one, second one. This is also third one. The peak gets higher and narrow. See here, the peak is head. We are getting higher and narrow nature. The peak shifted in the one direction. See that the peak shifted in the one direction only. This one. Third one is we will get the 2 peak forms. It exists in 3 forms according to their what is the natural number of individual with the performance. At the same time, they are indicating the phenotypical characteristics. Okay, these are all are very, very important things. See that. So another one is also given the same things also. The peak get higher and narrower. Peak shifted in the one directions. 2 peak. What is that? Forms. So this is the end of the part 3. Evolution is finished. And we will pass on to the next one is called as evolution part 4. Okay. See that. So do you have any doubt in this area, my dear students? Here, slightly just to go through it.
No,
Yes, just to go through it, then only it will be better. Once again, repeat for you. So there, what are the changes occurs? Where is the important questions is also there. Just note it down. Then only you can easily attend the question paper. Okay, last part, evolution, last part. We are going to study the human evolution. We are also one of the social animals, originated from the monkeys, isn't it? How many years? Do you know? Nowadays we are sending the rocket to the, which one where? Moon. Before that, we must know the origin and evolution of man. Okay. What are the phylum? Classification. There is nothing but what is that? Classification of man. Sometimes they may ask the questions. Very, very important. Okay. Which order they are asking? Order. Primates. Subclass. Eutheria. Like that. Already we know about genus and species. Already we know about because the binomial nomenclature Homo sapiens. Okay. See here. one by one, the classification of man, phylum, chordata. We are having the backbone. Subphylum, phylum, subphylum, vertebrate. Class, Mammalia. Subclasses, Eutheria. Order, Primates. Note it down, very, very important. That's why I have put a symbol like this here. It's very, very important. Which order? The human beings also comes under the order is called as primates. Suborder, Anthropodidae. Subfamily, Hominidae. This part also so many times they were asked. Family, Hominidae. Hominidae, Hominidae. Genus, Homo. Species, Sapien. These are the important things. You must note it down. Which one? Chordata, vertebrate, Mammalia, and Eutheria. Subclasses sometimes are Eutheria. Orders, primates. Okay. Family, Hominidae. That is very, very important. See here, the primates included prosimians. Prosimians, primates. Primates means here, the order. Okay. So many prosimians, lemurs, tarsirias, and related forms. And anthropodias, we are all comes under the which one, anthropodiae, monkeys, apes, and humans. Okay. See, they are descended from small rodent-like or insectivorous mammals that are evolved about 80 million years ago. Man or mammals, particularly, that evolved how many years? 80 million years ago. That has given here how the human was developed from the previous one to the modern one. It is given as a what is a equation like format. See here, Ramapithecus is the first one. After the development, Australopithecus and Homo habilis and Homo erectus. Okay, here which one? Homo. Here, and Homo sapien is the last one. See, so this is very, very important things. Okay, so this is a way the development of the human being from the evolutionary aspects. Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Homo. Here it's a and Homo sapiens and sapiens. These are the things. See, first one is origin and evolution of man. You must note it down. Man's place among mammals. Men's place among the mammals. We are all concerned with mammals, isn't it? The mammals evolved from the primitive or reptiles. That is the Therapsida. In the Triassic period, about 210 million years ago. Okay, these are all present in a book. Mammal existed as an inconspicuous group of small rat-like creatures for about 150 million years during Mesozoic era. This area only, this time only, what happened? The dinosaurs also present in it, isn't it? Diversification of the mammals had taken place after the existing of dinosaurs. Okay, see, already told you. You just note it down. No need to. Sometimes they may ask the question. Primitive reptile is called as which one? Therapsida. They may ask the question. Humans also arised from the one of the primitive reptiles. Okay, which one? Therapsida. Sometimes they may ask the question. Mammals formed from the primitive reptiles. Name is called as what is that? Therapsida. Like that. They may ask the question. Okay. And modern primates included man, great apes, and also monkeys, and Ioris, and lemurs, and also tarsirids, originated about 65 million years ago during the Cretaceous period. Okay, man also originated which period? Cretaceous period. Just note it down. It is very, very important. Or apes or monkeys also Cretaceous period. The anthropoids, apes, ancestor of monkeys, apes, and mans, originated about 36 million years ago from Oligocene to Miocene epochs. These are all the epoch periods. Okay, 36 millions. After that, what happened? The hominids, ancestor of apes and man. So this is our area. Apes and man evolved about 24 million years ago. 24, 12 add 36. Here, how much is that? You have to add 29. Again, 150 and 210. Okay. So this is very, very important. You must note it on 24, 36, and 65. Okay, like that you analyze, identify or remember that points also. The year is also very, very important sometimes, and also sometimes they may ask the questions. Evolution of hominids took place in Asia and Africa. Okay, so Asia and Africa. Suborder Anthropidae and also the Simons that include the New World monkey and Old World monkey, apes and mans. These are all are the, what is the mammals? How the mammals are derived, evolutionary aspect, how it will becomes, like that they may ask the questions. Okay, see, what are the general characteristics of mans? Once again, repeat for you. All the monkeys, okay, in ancestor, they will not erect posture. The only, what happened, they can use the 4 hands for the locomotory purpose. Okay, and another thing is very, very important. Skull also very, very difficult. And the eyebrow, okay, the mouth part, and also leg, tail, and also their fingers. Okay, these are the very, very important points when you are going to study human evolutions. See here, possesses erect posture and bipedal locomotion. What is the meaning of bipedal? Pedal locomotion is nothing but just when you are cycling. Likewise, you have to take just normal walking like that. Bipedal. Some of the animals, monkeys are, what is that? 4 pedal locomotions. They can use the hand also. That means front leg. Okay. Possesses large sized brain. That is very, very important. You must note it down here. The brain size is also vary. So many times they were asked the questions. Brain size, how much? Homo sapien, brain size. Okay. Skull shape. Like that they may ask us. Whether it's a posture or a erect posture. Bipedal. Like that they may ask the questions. Have a foramen magnum below the brain box. This is also another one. Foramen magnum is another part. Have a prominent chin with a prominent nose. Okay, take an example, monkey as well as a human beings. Both are having the same shapes, but the chin as well as which one, nose is different. Okay, monkey, they don't have the nose. I think it may be completely, what is that? Sunken compared to us. Okay, so the longest parental care. Okay, this is also very, very important. Ape as well as the human being, the parental care. Already you know very about it. Have a stereoscopic vision or 3-dimensional visions. What is my stereoscopic visions? Stereoscopic. When you are going to see any one of the long distance animals, we can easily see. At the same time, suddenly change the eyesight to the nearest one. That is called as stereoscopic as well as scopotopic visions. 3-dimensional vision. 3 dimensions means we cannot able to see the back sides. Only we can see the 270-degree aspects. That is 3-dimensional. Have a long period of postnatal growth. These are all very, very important. These are the characteristics of man. Okay. How the evolutionary changes that occur in humans are development of prominent chin. Chin also developed. Increase the cranial capacity. Reduction of brow and ridges. Already, there is eyebrow is there. Take an example, some monkey or other animals. Take even in the Ramapithecus or Australopithecus, that human beings also having a ridges, eyebrow ridges is also very, what is a thick in nature, prominent. Okay, and increase the cranial capacity, reduction of brow ridges, development of speeches. They cannot be able to speak. Only shouting the respective sounds. Okay. Murmuring. Okay. And also, what is that? They do not speak. Okay. After the evolutionary, after the identification of the letters only, the Tamil or English, whatever it may be, the letters only they can speak. Otherwise, they will not speak. Okay. That is also one of the evolutionary of development of community life. Community. Okay. Reduction in size of the teeth, very, very important. Take an example of monkey as well as human being. The teeth is also similar, but at the same time, nowadays, what happened, the evolutionary aspect, when we concern the evolutionary aspect, teeth size will be reduced. Reduction of muscles. Take an examples of chimpanzee. Okay. And ape. They are having a lot of muscles. Okay. Easily they will broke the which one, hardest material. Okay, with the help of leg or something else. And also teeth also very strong in nature. Gradually development of the thumb opposability. This is also very, very important. Thumb also. Development of shorter forelimbs. The fingers also shorter compared to the animals. Liberation of what is that? Hang to the locomotive function. Once upon a time, what happened? Hanging on the trees. Hands for the locomotive function. Slow assumptions and the pet pose. These are all very, very important for the changes, evolutionary changes occurs the human being. It was observed by the scientists and also reported here. Okay. See, what are the similarity between apes and man? This is very, very important. Here, you must note it down. General characteristics. Then evolutionary characteristics of man. Then comparison between which one, apes and man. That is, what are the similarities? What are the dissimilarities? Okay. Here, what happened? Just take an example. Absence of tail. Apes, it has a tail, but man do not have the tails. Broadened chest due to the flattering of sternum. Okay. So we are having the chest and also sternum also. So many bones are, it's connected with the sternum already in our but broadened chest. Smaller lumbar region. Lumbar region means just to take an example of backbone. When we are sitting, the lowest bone is called as lumbar region. Due to the reduce the number of lumbar vertebrae, the size of the vertebrae columns also very less or reduced or condensed. Okay, like that. Relatively larger brain and cranial cavity, efficient memory. Okay. The ape and man, they both are having the, which one, a large brain at the same time cranial cavity. What does it mean cranial? Where the brain is possessed, there is a small, what is a well-like structure that is called as cranial. Okay, efficient memory. Both are having the efficient memory. Okay, rememberable. Prominent brow ridges above the eyes. Okay, already ridges means the above the, about that iris. Okay. And capacity of communication by vocal means. Communicate with the language, its own language, isn't it? The communication is also very, very important. Highly developed facial musculature for expressions of rage, surprise, pleasure, and laughter by facial gestures. This is very, very important. Okay. Monkey also expresses itself. Apes also, man also express. Tendency to line in pair as a couples. It is very, very important. Human beings as well as the apes also tended to line in pair as a couples. Menstruation in females. This is also a common, but there is a, what is that? Which cycle? Oster cycles, where it present oster cycles in the cow or buffalo. But here, the ape as well as the man, they are having the menstruation cycles. Social behaviors. Okay, we are all are social animals. Likewise, the apes also social behaviors also present in it. That's why. And similarity in blood proteins and a banding pattern of chromosome. This is very, very, very important. We are having the chromosome 46, isn't it? Somatic cell, it consists of 46 chromosomes. At the same time, sexual reproduction cells consist of 23 chromosomes. Similar pattern present in it compared to ape as well as a man. 48 chromosome present in the apes. 2 chromosome is excess compared to human being. These are the similarities between the apes and mans. Okay, so this is also a very, very important. Okay. How the apes are identified means with the help of fossils. See here how many fossils and which year, where they identified which area, that things as given here. See, there are 4 type having given, I think 4 yes, 4. See, fossils of apes, Proplipithecus. Listen here, Proplipithecus. Origin and evolution in Oligocene epochs, so called as oligogenic epoch. Evolution about 30 to 35 millions ago. This is called as the first one. And Egyptopithecus. Origin and evolution in the late Oligocene. After the Oligocene, there is a Egyptopithecus and a Miocene. Oligocene and Miocene. After the Oligocene, OM, just to mention it. Epoch, so is called as Miocene epoch. Which epoch is mentioned here, that is the, what is that? Apes is also that particular ape mentioned. They are collectively called as that epoch, and apes also similar, Miocene. And proconsul, it is fossils were discovered by the Leakey from East Africa near Victoria Lake in Kenya from Miocene rocks. Miocene rocks means these times what happened, after the Oligocenes. There is one of the rocks also identified which area. This is very, very important. After Africa near Victoria Lake. It walked on its 4 legs, considered as a common ancestor of man and apes. We are all walking in the 2 legs, isn't it? Okay. Our ancestor, 4 legs. That is the difference. Okay. And then the 4th one is called as, what is that? Dryopithecus. This is very, very important. The evolution in 15 to 20 million years ago, the direct ancestor of modern days apes. Okay, the modern days apes also comes from where, which year, how many years? 15 to 20 million years ago. They were forest dwellers, spending most of the time on the tree only. This is also another one. See, Dryopithecus must you note it on which one, what are the general characteristic features, particularly tell us which times, which area, okay, which years. Okay, direct ancestor of modern days apes, and where it lives, okay, on the trees only. Origin and evolution is Miocene epochs. Semi-erect posture, they will not stand as it is, they will not stand straight, they just slightly bend in nature. Quadrupedal locomotion. Quadrupedal means, what does it mean? Slight. 4 limbs longer than hind limbs. 4 limbs longer. Okay, then hind limb. Hind limb means the back side there is a one limb is that limb is very short compared to the front one. Thick growth of hairs. Hairs already in our each and every monkey, they are having number of hairs compared to us. Some of the human beings also having so many hairs. U-shaped jaws. Okay. Teeth large and sharper. This is also another one. By nature, vegetarian fruit eater. This is very, very important. Dryopithecus, there is a, which one true or false, they may ask the question. Assessment reasoning also, they may ask the questions. Dryopithecus, the evolutionary period is called 20 to 25 million years ago. It is not a Miocene epoch. Erect posture or semi-erect posture. U-shaped jaws. But it is a, what is a vegetarian and fruit eater. Sometimes they ask the question, they are not vegetarian eaters. Dryopithecus is not a vegetarian or fruit eaters. Like that, they may ask the question whether true or false or match the following. This is very, very important. They are all are, what is that? False in nature. Vegetarian fruit eaters, Miocene epoch, semi-erect posture, only on the trees, and also 15 to 20 millions years ago. So these are the things you must note and see that starting from the Paleolithicus, that's a modern Homo sapiens. These are the changes. How many changes here? Count it 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15th one. We are the modern Homo sapiens. Okay. Paleolithicus, Proconsul, Dryopithecus, Oreopithecus, like this. Okay. So development of the solo man. Okay. See here, the erect posture, not like this. Okay. This is the off bend in nature and slight, and also another one. So where there is a correct structure, see that this one, airily Homo sapiens. This is the correct position, isn't it? After that, straight. Listen, these are the evolutionary characteristics. Before that, these are the primitive characters of the human beings. So these are all very, very important. Dryopithecus, this is semi-erect posture. See the structure of semi-erect posture. Okay, that things is here. You must note it down. Very, very important. The last one is called as Ramapithecus and Sivapithecus. Fossil discovered by Levis. Note it down from the Sivalik in Punjab hills in India. The Levis. Okay, Ramapithecus discovered in by where Punjab and Levis. The spelling mistake here. Okay, and India is also. The region where Ramapithecus lived was open grassland. It is another characteristic. It's not in the tree. The open grassland only. The hominids status for them is climbed on the two grounds. These are the characteristics of features of the Ramapithecus as well as the Sivapithecus. See, so this is the Ramapithecus structures. Okay, see the skull shape. Okay, these things is very, very important. See, Sivapithecus. See the structure. Only the presence of the grassland. Okay, that things also very, very important. So remaining things we will see the next classes, Australopithecus and how the evolutionary of the hominids and that things we will see. Okay, here what are the things we discussed? Will you please tell me what is the general characteristics of man and how they are classified? Okay, classification of the man. And Ramapithecus, Australopithecus, Homo habilis, and Homo erectus, hominids, and Homo sapiens. Okay, and origin and evolution, man's place among the mammals. Okay, that things also very, very important things. And what are the general characteristics and also evolutionary characters of the man? Also already discussed. Similarities between say apes and man. That things also we saw it. What are the types of fossils of apes? That things are three things: Prolopithecus, Egyptothecus, and the Prosonals, and Dryopithecus. Okay, and the last one is called as what is that? Sivapithecus and Ramapithecus. Who discovered the things? Levis. Where? Punjab in India. This is also very, very important. Levis. Don't forget it. Who discovered the fossils of Ramapithecus or Sivapithecus? You must note it on Levis. Where? Particularly Sivalik, Punjab. Note it down. Punjab hills in India. Okay. So this is the structures. Remaining things we will see on the next class, the Australopithecus. Okay. And prehistoric man. And then other things also we'll see the remaining things, remaining classes. Okay. Thank you so much for the participation and concentrate and go through it. Then only it will be better for you. What is that? Read the question, read the lesson once again. Okay. Thank you so much, Ba. Thank you so much.
Thanks, sir.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you so much.